Hello, I'm Simone Pedrini. I'm a research fellow at Curtin University based in Perth in Western Australia.
Forest ecosystems around the world are under threat, and no more so than here in the biodiversity hotspot of southwest Western Australia, with what was once one of the largest temperate rainforests. Myself and my colleague, who's going to present immediately after me, Dr. Simone Pedrini, have been working in the ecosystems of southwest Western Australia and concentrating on systems like the forest ecosystems that we're in here to try and understand how to put back not just the trees, but the biodiversity that are part of a fully functional, resilient ecosystem. And as part of this journey, we've understood and have come to appreciate that so much of the other animals and plants and the trees show a terrific interdependence, and that ecosystems such as this forest ecosystem, the Jarrah and blackbutt forests of the southwest of Western Australia, can be sustainably and in perpetuity put back together as long as we put back a full complement, or as best a complement of species as possible. Things such as this remarkable water bush, a legume, has a soil seed bank that potentially lives 50 years or more, is capable of restarting the whole nitrogen cycle in the system. The maidenhairs that cover the ground here, related to the European maidenhair, are also part of the soil stability. And the leaf litter that's created by all the combination of the up to 300 species that share the forest canopy with the great giant trees that we have here, are all part of creating that dynamic, resilient and climate-proofed ecosystem. But in that journey, it's important that we do get some of the fundamentals correct. And when we start moving away from just the trees through to the biodiversity, issues become important that maybe were not important with the tree species. And one of those, and one of the most critical, is the seed supply chain. How robust is it? How is it able to support the scale and diversity of the species required? And is there technology inbuilt in the seed supply chain to ensure the quality and diversity of diversity is put back, but also that the techniques for that restoration are in place? I'll now hand over to my colleague, Dr. Simone Pedrini, who is working on a range of projects to outline some of the issues and solutions to the seed supply chain.
So as Kingsley said in his forest talk, is that there are problems in the supply of native seed globally, especially when it comes to the quantity and diversity of quality of seeds. And there's not enough of them to fill the growing demand for native seeds globally in this push for restoration and reforestation. To do so, we decided to analyze the native seed supply chain across the world and decide to publish a series of standards to outline some of these problems. To do so, we've decided to analyze the seed supply chain in the broader context of ecological restoration, starting from an ecosystem to be restored and returning a restored ecosystem, going through a restoration plan, a native seed sourcing and procurement plan, and then starting from seed collection from natural populations and eventually seed production system, then going through seed processing, seed quality testing, if needed, seed storage, seed dormancy alleviation, and seed enhancement, to ultimately have seeds ready to be delivered on the ground and through monitoring and various activities, return a restored ecosystem. We've published this review of the native seed supply chain in this special issue published last year in the journal Restoration Ecology in a series of papers that provide an in-depth analysis of each of the steps of the seed supply chain. And it culminates in the international principles and standards for native seeds in ecological restoration, where we try to provide some solution to all of these problems. The issue with the native seed supply chain varies, and the main one, the first one, is that it's not clear what can be considered native seeds for ecological restoration. And then there are very few policies or certification schemes that can regulate these developing markets. And in most countries, there's usually very little consideration for quality of native seed batches. So going to the first questions, what can be considered native seeds for ecological restoration? The key aspects of native seeds is the population of origin, so where it comes from and how it was collected, because this collection needs to be representative of the donor population, and this diversity needs to be preserved as far as practical throughout the supply chain, so that it is maintained when delivered to site. This will mean that selected varieties, also called nativas, are not recommended for ecological restoration. They might still have a use in forestry, maybe in revegetation, in rehabilitation, but if you aim for ecological restoration, it's better stay away from improved varieties. This brings me to the key differences between selected variety for crops and native seeds. The selected varieties are the result of hundreds, if not thousands of years of plant breeding. So some of the traits, such as dormancy, seed fill, have been altered significantly to improve reliability. And the key information and the key attributes that need to be maintained throughout this supply chain is the genetic purity and the varietal stability. Native seeds, on the contrary, are representative of a broad array of genetic diversity, and that results in high variability and logistical complexities. And the key information that need to follow a seed batch is the origin of these seeds. But ultimately, the main difference between these two kinds of seeds is that the first one, the varieties, are aimed for the production of foods or goods or services, when native seeds are there to restore ecosystem. They should be resilient, and therefore the genetic diversity, it's there to guarantee this resilience of these newly restored ecosystems. Unfortunately, if this difference between varieties and natives is not understood or acknowledged by the policymakers, it does result in a series of regulation that, though well-meaning, might actually be detrimental to the development of native seed markets. And there's a similar example in Brazil when they try to retrofit forestry regulation to native seeds, and in Europe where they try to use fodder species regulation and apply it to natives for grassland. A similar issue is faced when seed testing is to be performed, because now the International Seed Testing Association has very strict and precise standards on how testing should be performed on selected varieties. But there's a little wiggle room to allow for the diversity of native seeds. So registered laboratories have trouble processing testing native seeds. And this has brought the perception in native seed supplier that the quality of native seeds cannot be tested effectively. And this is what we're trying to change with this standard document. We instead want to find a balance between what are quality expectations and guarantees for the users and what is practically achievable and economically viable for seed suppliers. When we talk about seed quality testing, we mean all those attributes of a seed batch that can be given a numeric value, such as purity, viability, and germinability. In this example, in the document, we provide this pro forma label that shows how, for example, seed quality testing could be reported. And if you break down the seed testing into its various steps, the first one is the seed purity test. So let's say we've got a 10-kilo bag of native seeds. We'll take a representative sample out of the bag and then run the purity test, which means separating what is considered, what is clearly a pure seed unit and what is non-seed, like twigs, little rocks, or leaves. In this example, we've got 90% purity. At this stage, it's also useful to record the weight of seeds and to have an understanding of how many pure seeds units are per gram. Once we have this pure seed unit, we can then run a viability test. There's a large array of tests that can be run. The cut test, for example, X-ray, tetrazolium, floating test. And ultimately, this test will result a number, for this case 80%, of seeds that can be considered viable because look healthy and filled. You can then run a germination test. Germination test is usually done on an appropriate substrate that allow water to be given to the seeds. In this example, we've got 50% germination. Then the difference between the results of the viability test and the germinability test will provide some indication on what percentage of the seed batch is to be considered dormant. In this example, 30%. Those information are useful, but to make them very meaningful for the seed user or the seed suppliers, they need to be multiplied by the purity test. By doing so, we actually achieve the most meaningful results, like the pure life seeds or the pure germinable seeds. And especially the pure life seeds information is the key for every seed batch. For example, now, in our 10 kilo bag, we know that 7.2 kilos is actually pure life seeds. And our suggestion for seed suppliers or seed users is to move away from the concept of talking about kilos of seed, and instead start using the pure life seeds as a way to market and sell seeds. So far I've talked like how we suggest how the testing should be performed, but we're also working on technology to speed up the process of seed testing and decrease the price of testing. For example, we're trying to develop a series of techniques using X-ray scanners, similar to the ones used in hospitals, to look inside the seeds without killing them. These methods already allowed us to speed up the process of seed testing a lot, but we're now moving into the space of developing an algorithm and a software that can help the testing and provide semi-automated results, so you can cut the time for testing a seed batch from half an hour to less than 5 minutes. Another key step in the seed supply chain is seed processing, which means the manipulation of a seed batch to improve the purity and its viability. It can be done on small scale, manually using sieves, or on a large scale, different kind of equipment. But then goal is the same, so to get a seed batch that is not pure and get perfectly pure seeds at the end of it. This is an example of some small-scale lab cleaning using a series of sieves, threshing, this can be manually or using a thresher or dehuller, and then using airflow, it's possible to separate seeds from other material and empty seeds using the difference in density. In this example, we can manage to achieve an almost 100% purity. This picture here, that's one of the eucalyptus that grows around Perth, and we go from a 7% viability to almost 80% at the end of the cleaning process. Once we got our seeds clean, we can then perform some seed enhancement. One of the seed enhancement is seed priming, when seeds are imbibed for a certain time and then dried up again. And this usually can improve the quality of all seed batches and improve synchronicity and speed of germination. But the technology I'm more interested in is seed coating. Seed coating can be simply explained as putting stuff around the seed. It can be very tiny, like film coating, a very thin layer of material, encrusting when you build up a bit more, or pelleting when the shape of the seed is no longer evident. It's been performed in agriculture for almost a century because it helps standardize the weight and the size and physical property of a seed, but also to deliver beneficial compounds such as pesticides, insecticides, nutrients, and phytoactive promoters, and so on. In this video, I'll show you an example of how I performed the pelleting on this Australian shrub. And you can see how drastically the size of the seed and the shape is increased through this process. Going back to the species I showed you earlier, this eucalyptus, you can see that we can go from these seeds to these seeds. So it's almost a 30%, 13 times increase in volume and 9 times increase in weight. And this is without adding any beneficial compound, but the just variation of the seed size alone can have great effects when restoration needs to be performed at scale. And I want to bring you the example of the Western Australian wheat belt. This is a very large area that has been cleared in the last 100 years for agriculture, so that now less than 7% of the wheat belt has this remnant vegetation. There's lots of projects now starting, try to bring back some of that bush, either for carbon sequestration or for biodiversity restoration. And currently, what is done on this land is to mix the seeds of different species, with of course different size and morphological complexities, with inert material, and then deliver them through this customized bit of equipment that can cover a maximum of 10-15 hectares per day. Now, if we manage to get all the seeds at a similar shape and size and weight, we can maybe then start employing already existing large-scale agricultural equipment and start covering the large-scale landscape scale of restoration and reforestation that is currently needed. That's the end of my talk. I'm not sure if I'll be available to stay for the panel talk, because I'm in a remote part of Australia and my internet connection might drop at any moment. If I am not able to answer your question live, you're welcome to send them at this email address. Thank you for your